Thank you very much for the invitation to speak. I'm going to take slight liberties with the topic I was assigned of imaging to give a little bit of an update on where the sort of brain atlasing is going, but with more of an emphasis on moving beyond these sort of ground-up single-cell suspension classifications to trying to map these transcriptomic types onto tissue and to try to correspond with other types of features. And I should also say that the collaborative spirit that we're seeing throughout here is rampant on the brain side as well. We have now several big new projects that are engaging many different members of the brain community to work together and try to accelerate this field. So I should say that at the Allen Institute, although we have a reputation for brain atlasing, our sort of forays into single-cell analysis have actually come from a slightly different perspective, which is treating the brain as a circuit, and to try to reverse engineer the brain by understanding its cellular components and the connections with one another, somewhat akin to thinking about taking an electronic device like a cell phone and trying to understand what its parts are, how they connect with one another, and how this actually turns into a functional circuit of some sort. So for the last 5 or 6 years, we've been sort of engaged in a big effort to try to systematize this approach, in particular in the neocortex, both in mouse and human, where one element of this is the cell type element of can we understand what all the different types of cells in a local domain of cortex are, based on various different data modalities and hopefully the intersection of those modalities. Once we start to understand that taxonomy, can we understand how they actually form a circuit, what the properties of connectivity are among those? And it also turns out that by having a molecular focus on the cell typing, this leads directly to the generation of cell type specific tools, which we're trying to do both as transgenics in mice, as viruses in human. And hopefully, eventually, we can start to build models based on this data and merge the tools and the models to test predictions of some sort. This first part is really where this intersects most strongly with the human cell atlas idea. And in some sense, I think that this can be sort of a prototype for where the atlas goes in the future, that initially you want to sort of define the parts, but that's not the end of the game by any means. Then you want to understand what those parts are and what they do functionally. So I also wanted to make another point here, which I make at every one of these meetings, which is that an atlas is a work of anatomy. And it's really important to keep track of that anatomy. In the brain, of course, for many years, reference atlases have been part of the suite of tools that we work with. This is certainly the case here, and I think it's really important to think for each of these organs that you are very careful about the precision with which you map the specimens you're getting, because location is extremely important in defining the function of these cells. And so for applying to the brain, we both keep track of the position so that we can map it back into a common coordinate framework, but we also pay very strong attention to the precision of the dissections in the first place. And so, for example, with the piece of cortex, this would start from a whole brain that's slabbed, dissect a region, isolate that region, we can stain it, we can isolate individual layers or subnuclei of regions, and do each of those independently. So we actually have a lot of positional information up front as we go into this. And what we've been doing for the last few years then is to start to apply single-cell transcriptomics to these samples, and really actually to try to saturate in these areas as best as we can. That unlike sort of many of the sort of initial surveys, we've taken a different approach, which is to go in one region and try to get, see how deep we can go. And so we've done this in both mouse and human. I'm showing the human version here. Human, we were actually forced to go to nuclei. So it turns out now lots of people are doing nuclei, and this is advantageous for certain things. It's necessary in the adult brain because you can't actually isolate intact whole cells or whole neurons. They don't survive this process. But the good thing is that we've actually shown you can get very comparable results with cells and nuclei. This is sort of the final result from a single cortical area in the middle temporal gyrus. I represent this more as the sort of periodic table rather than a TS&E plot. But we can actually distinguish many different types of neurons and non-neuronal cells, in all about 65 different types of neurons. And this organization, the tree structure that you see is extremely consistent with everything that's been done in the past, with the developmental origin of different types of cells, but it goes a level deeper than that as well. And so we have a lot of different kinds of cells now to try to think about the function. And I just wanted to point out a couple of sort of themes that have emerged from this. The first is that the complexity is very high, but it really is finite. If you do more cells, you just don't get more types. Eventually, you sort of saturate on this. And we've really shown this mostly in the mouse, where at initial taxonomy, we did 10 times the number of cells. You only see about twice the number of types, and it really is sort of saturated. So that's sort of nice that there is sort of an answer with this type of assay. Importantly, for the future sorts of assays, cell types are almost never defined by a single gene. These are combinatorial profiles that define different kinds of cells. And here, this plot is showing the frequency of the different types. And what's sort of interesting to me is that once you get down to this fine level of granularity, almost all types are rare, actually. So there are only a few types that are sort of dominating when you're looking at fine types. So this is actually from the perspective of defining a circuit or defining an organ. It is a really outstanding sort of organizing set of data. So unlike a sort of unbounded field in the past where you're trying to define based on morphology or other less quantifiable phenotypes, now we get an answer. We have a certain number of discriminable, robust types. And now we can ask, well, what are these things, right? So this is just the beginning. So there are biases associated with the original isolation, so we don't actually know what the proportions of these things are with great fidelity, or their spatial organizations beyond the initial dissections, whether there are other sort of features of gradients and things like this. On the flip side, we also really don't know how this relates to the properties of these cells. What is the anatomy of these cells? What are their physiology or functions? Who do they connect to? We don't have that information based purely on the molecular side. And so, this is a good start. Now we need new methods that allow us to correlate those things, to take them into tissue sections and to map their distributions and to correlate them with other features of cells. So the sort of success of this has led us to want to do a lot more of this type of thing just on the sort of basic atlasing. But the problem is that, especially in human, these are extremely difficult problems. And they're really kind of too big for individual labs to take on. And so we've started to make collaborations with different consortia of experts across the field. And so we've, in particular, started to work with Sten Linnerson, who all of you know very well, starting to look across many more brain regions with the transcriptomics, and to start to really look at the properties of these cells, which can only be done in living tissue sections. And so we've sort of teamed up with a small group of people that do living cortical resections from epilepsy surgeries or tumor removal surgeries to start to do these correlations. So this is one of the collaborative projects that I was referring to. And essentially the elements of it are to begin to move towards a whole brain atlas in human using single-cell transcriptomics. This is directly collaborating with Sten. So we're helping to supply the samples to do this, but we're sort of taking a first pass, which is about 100 different regions, and relatively shallow sequencing, but to give kind of a survey across the whole brain. We sort of opted for what I heard referred to as 2014 technology of Eve, which is plate-based, smarter whole transcriptome sequencing, which we still feel gives the most information per cell. And so sort of combining these with a targeted analysis in regions where we can get living tissue with a broader survey, and then moving into this sort of multimodal characterization that takes advantage of neurosurgical specimens, trying to link that with the transcriptome. So this correspondence is extremely challenging in human, but we've managed to set up now a rather robust system where we work with local neurosurgeons in the Seattle area. Our collaborators work with local surgeons in their local domains. And in the process of treating intractable epilepsy, for example, it's necessary to core through part of the distal cortex in order to get to the site of pathology. And so we're actually able to get these sugar cube or so size pieces of cortical tissue that are quite reproducible, section them as you would a model organism's tissue, and then do slice physiology on these. And so for some time we've been actually standardizing this. We have a lot of single cell recordings and morphologies on our website already. But in order to make this correlation to the transcriptome, we've had to move to a new technique, which is to use patch-seq. And many of you may be familiar with this. This is the intersection of patch clamp physiology and RNA sequencing. And so essentially, as I've just described, it's patching onto a cell, recording and filling the cell. At the end of this, you extract the cytoplasm, or actually what turns out to really be the winner is to extract the nucleus. This is a very resistant package of RNA that you get quite a consistent amount per cell. And so at the end, after you have injected current to get the firing properties of these cells, you've injected biocytin to get the morphology, now you get the transcriptome, and then you map it back to the original classification based on dissociated cells. So this is really powerful in that every one of these, we've now been able to pin 2 additional data modalities onto that original classification. And we're sort of trying to march across this to get representation across this whole thing. Now I should say, this is a 1 cell at a time effort. So this is extremely labor intensive to try to annotate this classification. So really, I think we need a different suite of methods, which I think almost every talk has mentioned at some level, which are the spatial transcriptomic methods, where you can look at highly multiplexed assays in tissue sections. And one of the uses of this, I think, that's most salient for this atlasing type of effort, is to move from this classification to define rational gene panels that can discriminate among the different types of cells that you see, and then map them back on the tissue section. So in one idea that you would get sort of a census of cell types with their accurate proportions and spatial distributions. On the other, of course, you can imagine moving this into looking at healthy versus disease or any sort of other variable on top of this. So we've been trying to work on this for some time. I'll get to there's a large community that's been working on this as well. These types of methods are becoming increasingly robust. This is a typo here, sorry. Hybridization chain reaction, for example, is an amplified method that works very well for nice robust markers. And this is in the mouse neocortex here, but just to give an idea of why we need these sorts of things, there are all these different types of cells defined by different genes intermingled with one another. There's not a spatial organization beyond the laminar organization. So you really don't know which of these is which without having that kind of information. Now I should say, on almost everything, working with human tissues provides additional challenges, and that's certainly the case with this as well. Since this is fluorescent imaging, problematic issue with adult tissues in the brain is that they accumulate these autofluorescent lipofuscin granules. And these things have very broad spectrum autofluorescence, and they obscure a lot of signals. So this is actually kind of an unsolved problem in the field right now of how to deal with this in combination with these sort of spatial assays, either experimentally or on the back end computationally. Now the other sort of obvious application of these methods is to use them as a post-hoc assay, where you've done some other functional assay or anatomical assay, and you then want to pin a molecular identity onto that cell type. So you can imagine you have lots of cells that have been filled with some genetic or other technique, or you're doing more complicated electrophysiology, or you're doing even calcium imaging, where you get functional measurements on many cells, and now you want to pin the molecular identity on that. So that's a little bit harder for the sort of thin section approaches. And just to give an idea that this is a real-world use case here, one of the other projects that we have going in human tissues is to try to start to understand the properties and the frequency of connections between different types of cells in human cortex. So we've established now a multipatch system where we have an octopatch approach where we try to record from 8 cells simultaneously, and then we can stimulate and record across this whole set and see who's connected with one another. But at the moment, the best you can do is to look at those anatomies and try to understand what kind of cell it is. We really want to understand what molecular type that is. So to that end, we've started to work with cleared thick sections using these multiplex methods. On the left here is an HCR-based approach using, I think, 3, maybe 4 markers in one round. It turns out with passive clearing, you can get good penetration of the probes into the tissue, so it's actually not necessary to resect this in order to identify, at least on a small set of markers, what each cell is. Now, it turns out also that we can combine that with the physiology, where we can put in a dye into a cell that we're recording from, and subsequently do the fish on the backside. And so I think we have all the pieces now that we're starting to be able to actually directly correspond these properties by doing this fixed section in situ hybridization on the back end. So I think, as all of you know, there actually are many quite remarkable methods that are in the field right now for doing the spatial transcriptomics. These range from the sort of simplest versions are fluorescent in situ hybridization done in sequential rounds of stripping and rebinding, through seek fish and mer fish, which get into more complex barcoding and spatiotemporal barcoding, to actually direct sequencing in a biased or unbiased way on the tissue section itself. And so it's actually a little bit bewildering to know, like, which method should I use and which method is best applied to this and has particular strengths for certain types of assays. And so with this sort of in mind, we're extremely fortunate that the Zuckerberg initiative was interested in trying to fund a consortium approach to accelerate the development of these methods and their application to this particular problem, which is taking a transcriptomic classification and trying to map these onto a tissue section. So I would spend the rest of my time naming all the people, but as you can see, it's a group of luminaries in this area, the Space TX Dream Team. So we felt that this group needed a flashy name, so it's like SpaceX for spatial transcriptomics. It's the Space TX group. And the idea is that for all of these groups together to directly benchmark their methods to one another with this particular problem, both in mouse and human. And I should say that, as I mentioned, sort of collaboration is in the air. It's really been remarkable that an additional group of people on the computational side have wanted to join pro bono to work with us on these problems. And I think that's sort of indicative of everything we're experiencing here in the Human Cell Atlas. So to be just a little bit more specific, what we're trying to do in this is the Allen Institute has generated two very exhaustive data sets characterizing cellular diversity in the human temporal cortex and in the mouse visual cortex. Additionally, this project will develop a prenatal visual cortex classification. And so we're trying to work from those to develop a sort of consensus classification and understand how best to derive panels of genes that can discriminate among those. Everyone's going to run their methods using those genes, and then we're going to try to compare all these together at the end and understand sort of whether we can recapitulate that classification, what the biological answer is, and sort of what the relative strengths of the different methods are. So to sort of organize this is sort of a tricky thing to try to organize. We've, I think, done some similar things to other projects here that we've formed working groups to tackle these different problems. So for example, we have a group that's trying to apply a variety of different classification and clustering methods to the data sets. We have a different group that's working on methods for probe design to optimally discriminate among all these types. We're hoping that the imaging and image analysis elements can also yield some standardization of methods and data standards, as well as a sort of a mapping back to these original classifications. So we're a quarter of the way through this project. This is only a one-year project, and the computational arms have already made quite a lot of progress. I think one particularly good piece of news is that we've had various groups run their different classification methods on the same data set, and we get very concordant results. So it's not method dependent. On the gene panel selection side, we're starting to get a really good understanding of what makes a good gene panel and how many genes you would need to be able to discriminate among many types of cells. And it's sort of a little non-intuitive, perhaps, that most people would think you should pick your best set of individual marker genes, but you can get very good results also using a totally combinatorial set that none of which look like good markers, but they give good binary sort of expression that gives a good readout. I should point out that one idea running around in the field is that a random set of genes would give a comparable result. And at least in our simulations, that is not true with a small gene panel. So it actually doesn't matter which genes you pick. OK, so next year, perhaps we can present on the actual results from this. I just wanted to end, I know I'm just about out of time, one interesting biological thing that's come out. So as I mentioned at the beginning, we've been approaching this classification both in mouse and human. And now we have the data to start making these comparisons. I think our sort of egocentric view would be that we must be more complex than a mouse. But it's actually turning out that the diversity of types with various caveats noted, does not seem to be particularly different. So there are, of course, some differences where you see expanded neocortex in certain layers, you see a bit more diversity, but the sort of take-home message is that the diversity is quite similar. So this was kind of a bit of a surprise, although it does get difficult to directly line up these very fine subtypes. But I don't want to leave you with the message that things are the same, because the details are turning out to be very different. Many of these genes that are most distinctive for particular types of cells have changed their cellular expression patterns between species. And this is not a minor effect. We see this again and again and again and again. And certainly the gene panels that you would pick to use these spatial assays will not be the same between mouse and human. So I think really this sort of highlights why we need to do human work in the first place. The details are not the same, even if the general organization is quite conserved. And with that, I'd like to thank a lot of people that have been involved in this. We operate in this big team environments. Also the funding organizations that are funding these nice collaborative projects that we're starting. I'll just leave with some of the people leading those projects. Thank you.
Okay, we're running a bit late, so just time for one quick question. Okay, then if there are no questions actually, then we'll just,
I think that was incredibly smart of you to stick with the SmartSeq on the nuclear profiling. So I think a lot of people are extrapolating, though, that hey, it must work as well with 10x to do the nuclear profiling. Is that, what's the status of knowledge around that?
I would say that those direct comparisons are happening in several venues at the moment. We've begun to do them at the Allen Institute, and I guess to be somewhat glib, you can get a somewhat comparable classification result with 10x the number of cells using 10x genomics. But it's still not clear that you got the total information content with that. So